Hello, everyone. Thank you for attending today's virtual roundtable, Challenge Accepted, Biopharma Puts COVID-19 in its Pipeline, sponsored by Patheon by Thermo Fisher Scientific. I'm Amira Al-Idris, an editor at Fierce Biotech, and I will be moderating this session. Our speakers today are George Scangos, PhD, Chief Executive Officer of Vir Biotechnology, Zi Hong, PhD, Co-founder, President, and Chief Executive Officer of Brii Biosciences, Anne Lean, PhD, co-founder and chief scientific officer of Allovir, and Amanda Bossi, president of Drug Product Division North America Pharma Services, Thermo Fisher Scientific. You can read their full bios on the left side of your window by selecting the Speakers tab, and here are just some technical notes before we begin. The session is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. The session is being recorded and will be available on demand within 24 hours after the event. Time permitting, we'll follow the presentation with a Q&A session. Please submit your questions using the question and answers tab on the left side of your screen. Before we begin our discussion, I would like to introduce Amanda Bossi, who will be providing our opening remarks. Amanda, please go ahead.
Thank you, Amira. Good afternoon, and thank you for the opportunity to open today's webinar. I hope that all of you and those that you hold dear are safe and healthy. As Amira said, my name is Amanda Bossi, and I serve as the president of Drug Product North America for the Thermo Fisher's Pharma Services business. We specialize in drug development, manufacturing services, as well as transportation management and clinical trial solutions to pharma and biotech customers of all sizes around the world. Before we start, I'd like to recognize the tireless efforts of the healthcare community that has continued working and making all efforts to serve their patients despite the circumstances. That includes the pharmaceutical innovators on today's panel and our audience listening to this webinar. I'd like to thank this community for stepping up to the plate to find solutions to the challenge that's in front of all of us. Whether you're in academia, biotech, large pharma, many of you are working around the clock to find a treatment for COVID-19. At Thermo Fisher, we're also working to not only respond to the prevention of spread, but also in treatment. During this time, our priority has been to ensure that our employees are safe while we maintain business continuity. And our customers can continue to develop and manufacture and deliver the medicines to patients that rely on all of us. We're working closely with the local governments and suppliers to navigate any local imposed quarantines or restrictions so that our sites can remain operational, given the importance of the healthcare system and our role in supporting COVID-19 response. As an example, in Italy, we've continued to manufacture a product at both of our sterile sites. We've also been able to successfully navigate quarantine zones to continue delivery thanks to Total Transportation Service Line. From a production perspective, our sites globally remain operational and ready to support development and commercialization efforts in vaccine development and production. We fully understand the need to get medicines to patients, even during extreme uncertainty. As you may have also heard, Thermo Fisher is helping in the battle with diagnostic testing. The company recently announced that the US FDA has issued an emergency use authorization for its diagnostic test that can be used immediately by laboratories in US to detect the virus that causes COVID-19. And we've also received the CE mark in the European Union. These are just a few examples of the roles that we're playing in battling the spread and also finding treatment for COVID-19. In these extraordinary times, we say that our mission to enable our customers to make the world healthier, cleaner, and safer couldn't be more relevant. In closing, I want to reiterate what an honor it is to be a part of this webinar today. Despite the uncertainty around us, there's a strong sense of community and purpose as we work in our personal and our professional lives to get through this challenging time together. This is specifically evident right now as you have all made the commitment to join today's session. It's a great testament to our commitment as a community to work together and to make a difference. So thank you. And back to you, Amira.
Thank you, Amanda. So it's time to start a discussion. We're going to break this down into 3 areas, starting with what kinds of treatments are in development for COVID-19, the challenges to developing and deploying those treatments, and then we'll talk a little bit about outbreak response and how that's changed over the years. So to start, what's in development? Maybe, George, you could start with what Vir is working on, the different types of ideas.
Sure. No, happy to do that. We actually have three approaches that we're taking to try and do something about this pandemic. First are development of antibodies that are neutralizing against COVID-2. We, as you may know, we have previously, one of the basic technologies of Vir is to go to patients who recovered from a disease and be able to screen a large number of patients and a large number of B cells from each of those patients to identify antibodies with desired properties, even if those antibodies occur rarely. And of course, you want potently antibodies that bind incredibly tightly, that are potent neutralizers, that are not strain specific and are more difficult for pathogens to escape from. So we were able to do that first with Ebola. And so one of the two treatments in the combo was a Vir isolated antibody, MAB-114. And we have a very interesting pan-flu antibody, recognizes all strains of flu A since the 1918-19 pandemic. All the pandemic strains, all the seasonal strains. That's now in a clinical trial, which unfortunately is delayed now due to COVID, to test its ability to provide a higher level of protection from flu, and also has potential for flu pandemics. After the SARS epidemic, we had isolated antibodies from SARS patients. We had characterized those antibodies for their ability to neutralize other coronaviruses, some of the other coronaviruses that are circulating in humans, as well as some of the different strains coming from a variety of animals. Once this epidemic started to become evident in January, we went back to SARS patients and isolated additional antibodies. And we now have some antibodies isolated from SARS patients that are potent neutralizers of COVID-2. So, and we are bringing those forward. The first of those are in manufacturing, and we would hope to start clinical trials this summer. We have introduced some alterations in the FC region that we think can enhance the activity of those antibodies. They have a mutation that extends the half-life that we've introduced into our flu antibody, and that has been used in some of the broadly neutralizing HIV antibodies that have been in humans. So we think we can protect patients for a longer period of time. And in one version of our lead antibody, we have an additional mutation that in animal models alters the way that the antibodies interact with other cells of the immune system, and both increases, for example, in a model of flu, increases the potency of the antibody about 5-fold in the near term, and over the long term results in the induction of potent T cell responses that protect the animals after the antibodies are gone. So our lead antibody, we have two versions. We'll take both into the clinic. One has that mutation, one doesn't. And of course, we have other antibodies coming behind that we think would be interesting to combine. And we think we have excellent antibodies. We are also in discussion with other companies to bring in yet additional antibodies. So the antibodies are one approach. We have a collaboration with Alnylam that's focused on siRNAs. And we have the lead molecule, which is an siRNA targeting hepatitis B that's in phase 2 testing now. And we have expanded that collaboration to include COVID-2 coronaviruses. So Alnylam has spent the past few years developing a lung delivery system, which they think now works well. And they are synthesizing a collection of antisense constructs with the potential to inhibit both COVID-2 and potentially be fairly broad in inhibiting coronaviruses in general. And so we'll test those, and should any of those work, we'll move them forward quickly. And the third approach we're taking is to identify cellular genes, host genes, which when inhibited or activated can interfere with the ability of COVID to infect cells and replicate. And we're doing that with a combination of CRISPR screening and high throughput informatics, so data sciences and machine learning. We've applied that technique to other respiratory viruses. We've gotten a very interesting collection of potential targets out. Sometimes those targets have already been targeted by some companies, not for the infectious disease application, but for other applications. And if we can identify such genes which already have compounds targeted against them, then maybe we can repurpose those compounds quite quickly and lead to some benefit. So that's our approach. A lot of other interesting companies on this call with similar and sometimes overlapping approaches and distinct approaches. So we need everything we can and all the approaches that we can to be taken forward by multiple companies so that we have the maximal chance of being able to do something to eventually end this pandemic. So I'll stop there, and we can turn it over to the next speaker.
Great, thank you. Thanks for mentioning all the proliferation of different approaches that the industry and academic groups and government groups, et cetera, are taking on. I know that Bree is also working on antibodies, but also in partnership with academic institutions. So maybe Z, you can touch on that before we pivot to Anne and your work with Alovir.
Well, thank you. Before I actually talk about what Bree has, I just want to quickly, what is out there. I think when we look, what are the opportunities out there, and then in the near term and long term perspective, we can quickly agree on some of the small molecule opportunities where mostly built on repurposed molecule. Those are the example of those molecules, like the Gilead's remdesivir and also the well-publicized chloroquine and hydroxychloroquine, where it was approved in development for other purpose and then indication that now repurposed for this. So that's one near-term opportunity that obviously that's out there. I think the other one that I think is also there near-term is the so-called plasma therapies. And those are the plasma that collected from a convalescent patient. Actually, it sort of lends to the validation of the neutralizing antibody that George was talking about. It, clearly validated in the recent Ebola clinical study and the PALM study, where demonstrating a number of the antibody from Regeneron as well as from Vir and shown to be effective against Ebola. There are obviously vaccines out there. So let me just tell you a little bit about what we have, and then what we're trying to do is trying to figure out a way how to maximize the opportunity, and see where we can actually contribute. So as you mentioned, we announced actually today a partnership with Tsinghua University as well as the Third People's Hospital in Shenzhen in China. And those investigators at the earliest time in January, they were able to isolate fully human neutralizing antibody from convalescent patients. These are antibodies specifically target COVID-19. So this is one opportunity we're looking at, and then with a near-term implication that we're moving very rapidly to the clinical testing in the near future. We have also recently announced a collaboration and then funding into Columbia University, and this is the effort led by David Ho. At Columbia, they're having multiple programs, and including small molecule program, and such as protease, as well as RNA-dependent RNA polymerases. The reason we're interested in those programs is we believe a very long-term, implications. We all know that all the recently repurposed molecules, they are not ideal. Either they are given IV infusion or they have many other side effects that are associated with. So it's our belief that if we want to take on coronavirus as a threat in general, we need to figure out whether or not we can develop a molecule in the long run that has the pan-coronavirus activities, and that can be easily given and easily scaled to both treatment as well as prevention of the coronavirus outbreak. So those are the programs we are also interested in investing in the long run. And that's the current approach we have. Obviously, we're looking for other opportunity to invest as well.
Thank you. I'm glad you mentioned a pan-coronavirus activity as being important in something new that we're developing. I know multivirus targeting treatments is something that Alavir is working on. So Anne, do you want to weigh in on that?
Yes, thank you very much. So Alavir is a company built around the use of virus-specific T cells as a therapeutic modality. And in collaboration with Baylor College of Medicine, we really have extensive experience, greater than 20 years, more than 300 patients treated with various iterations of virus-specific T cells, all targeted to different viruses that are potentially devastating and lethal in immunocompromised patients. And the reason we felt that it was important for Alavir to become involved in the fight against COVID-2 is really based on emerging data in the literature now demonstrating that a potent T-cell immune response is very important in protecting and fighting against the virus in individuals who are able to control this virus on their own. So with this background evidence that the T-cell arm of the immune system is very important in protecting and treating the disease, we thought it really fit very appropriately under the umbrella of what Alavir does and does extremely well, which is generate banks of cells that can be used as an off-the-shelf therapeutic. And although our major focus here, of course, is to develop a potent therapy targeting multiple antigens that are immunodominant and immunogenic to T cells in the context of SARS-CoV-2, we are also extremely interested in exploring the potential for the self-same product to be active against multiple different strains of coronavirus, because although we're extremely mindful, the problem that we're solving and need to solve today is this current pandemic, but other coronaviruses are incredibly dangerous and particularly problematic in immunocompromised patients, and that's certainly something that we are looking into in the context of the company. But in general, I think what you've heard today from the panel so far is the various different approaches that are being applied, and I think it's really important that every company here is bringing to bear their expertise in different areas of science and medicine and immunology to develop potent therapies. And I think there's really space for all of us to be able to bring forward the type of science that we do best. And certainly in Alavir's case, it's implementing T-cell therapies to treat infections in patients. And just a final point about T cells in terms of the modality itself. T cells can be infused to patients. They expand in vivo, so they do grow in patients once administered, so they can home to sites of disease and effectively eliminate infected cells. And so we really do feel strongly that this approach has a potential to make a real difference in patients with underlying risk factors that are associated with poor outcomes.
Thank you. I'd also like to thank Z earlier for bringing up the other treatments that are in development. I understand that I have a panel full of speakers working on brand new treatments, but I'd like to discuss a little bit kind of why somebody would try to repurpose a drug that had already been approved or been developed for something else in this pandemic versus why somebody would choose to make a brand new drug. Z, you talked about near-term and long-term goals. Maybe you could talk about like some pros and cons for both strategies.
Well, thank you. Yes, from the near-term, obviously, is the COVID-19 infection is more than just the infection itself. It's the disease behind itself, and also different people actually have different underlying diseases. There are ways that you can think about how to treat the diseases rather than just the antiviral and treating the viruses. I think for that specific reason, we have seen other approaches such as We've seen that interleukin-6 antibodies are being tested as we speak. I think some of the challenge with that repurposing effort is that, number one, I think these molecules are not really designed specifically for COVID-19. So therefore, there are certain product properties may not be perfect. For example, the formulation may not be perfect. For example, the AE profile may not be perfect. The efficacy may not be potent enough. And if you talk about targeting the disease of cytokine storm, and you can't really assume everybody having COVID-19 has the same kind of a cytokine storm. And some of this inflammatory response is actually important for eliminating the viruses. So it's really, if you're not careful, you could potentially harm patient. So it's that challenge in terms of find out who is the right person to treat and when is the best time to treat, and then to treat with what. That's always going to be a challenge in terms of when we talk about the repurposed effort. I think in the long run, and clearly, I think you want to have a vaccine. I think that works if you want to go out against this particular outbreak. If you can come up with a universal vaccine, and that would be great, but I think that would be very challenging as we learn from the experience with the flu. But from a small molecule, definitely there's absolute precedence in the past in other virus diseases where you can develop pan-viral and RNA-dependent RNA polymerases.
Z, I can't hear you. Can anyone else hear Z? No, I think we lost your audio, Z.
Can you hear me?
You're breaking up, G.
Can you hear me? I don't know where I got dropped off.
Yeah.
I can hear you guys.
Uh-huh. Okay.
Okay.
Well, maybe I can jump in here, and
Yeah.
because look, I completely agree with G's comment. They're right on the money. I think you can think about this, I think, in a number of different ways. Like, let's take whatever we have now that might work and try it. So if you think about chloroquine, azithromycin, and remdesivir, those are existing drugs with some reason to believe that they work. I mean, chloroquine has a couple of negative trials, a couple of positive trials. Don't know. I think it's clear if it were a home run, if it were curing this disease, we would know it. And so the question is whether it has some ability to slow the disease in some patients. And those trials, I think, that will definitively answer that are going on now, as they are with remdesivir. And so those are attempts to just do what we can in the near term to try and improve outcomes to some extent, right? Then there'll be the next generation, and G has antibodies, we have antibodies. Whether those first antibodies will work and whether they'll work as by themselves, whether the combos will be the next thing that we'll learn. And eventually we'll get to combinations of antibodies that really do provide very effective prophylaxis and treatment. I believe that, and we probably will get to small molecules that are very effective at controlling the drug as well. And as G was alluding to, and I think he started to have audio problems, we need a vaccine. And we need really effective vaccines that will be a major part of controlling this disease over the long run. And Anne's work, which is, I think really could provide substantial value to patients who are critically ill and whose immune systems are inadequate to fighting the disease. I think all these approaches will come. We need them all, and they'll come over different periods of time.
Anne, would you like to add to that?
Yeah, I completely agree with George. I think, with all these different therapies, we're all trying to deal with the pandemic, and it may turn out over time as we explore these different modalities that particular approaches work better in certain subsets of patients. At the moment, I think it's incumbent upon us to bring forward what we have and test it in the context of clinical trials and move as rapidly to patient care as possible. Again, specifically, I think with these targeted agents that you're hearing about today, I think the hope is that we will provide patients with benefit, and we may see benefit with all these different modalities. Ultimately, there's a need for preventative agents, there's a need for therapeutics, and then there's specific needs for therapeutics that deal with particular patient types that have underlying risk factors that may be served by one therapy more than another. But in general, I think these therapies all have a place and potentially could be combined or used sequentially and so on. So I think what you've heard from all of us is that our therapies are not mutually exclusive and quite complementary, in fact. And I think that's something that also should be kept in consideration.
Thank you. So moving on to the next piece, which is challenges to developing these treatments and then deploying them. There's a few that we identified. One of them, a big challenge, and I know a couple of you can speak well to this, is manufacturing, especially for a small company. So maybe, George, you want to talk about Veer's example?
Sure. Look, if you think about manufacturing antibodies, then you have to have that manufacturing capacity lined up before you know the outcome of your clinical trials. Or else you're going to finish the trial, and in the best case, the trial works and you have some drug. If you start manufacturing then it's months before you make that trial available to patients. And aside from absolutely being the wrong thing to do for patients, it would absolutely be a PR nightmare for the company. So you have to start preparing that manufacturing capacity well in advance of knowing the outcome of your drug. And of course, that takes capital, takes money to reserve that manufacturing capacity and to pay for manufacturing. And so it's not for the faint of heart, and it takes a company that has adequate access to capital, or and is willing to put some of that capital at risk by starting to develop the antibodies before you know the outcome. And so we're doing that. We have collaborations with WuXi. We have collaborations that we're finalizing with Biogen, both of whom are already working on our lead antibodies. And so we hope from that to get a supply of antibodies so we can be clinical testing, as well as cell lines that can be scaled up to put into the large capacity plants that exist around the world. So we are, that's what we're doing. I think it's a challenge for all of us making antibodies to access that capacity, but I think the manufacturers also are understand the urgency and are willing to work cooperatively with us.
Yeah, and Anne, do you want to jump in there? As I know, manufacturing is kind of a big bottleneck in cell therapy.
Yeah, I think that's really the perception for many T-cell-based therapies. I'd like to highlight a few differentiating components of AlloVir versus others. So our platform really is built for scale up and mass production to be able to accommodate something on the scale that's going on currently. And so it's really a platform technology to generate large numbers of very specific T cells, multiple doses, hundreds and thousands of doses, in fact, for patients. And these cells are generated in advance clinical need, as I mentioned, from donors who accommodate the broad patient population who could benefit from such a therapy. And they're maintained as an off-the-shelf reagent. In terms of how we're doing our current manufacturing, as we do have a number of products in development now and poised to enter phase 3 clinical trials, we're actually working and have done the tech transfer to a contract manufacturing facility. And so that's already an ongoing body of work to take forward our virus-specific T-cell therapies for other latent endolytic viruses. But also, I would like to highlight that Alavir is a company associated with Elevate Bio, a portfolio company of Elevate Bio. And Elevate itself is really a company dedicated to the advancement of cell and gene therapies, so much so that they're in the very late stages of building out their own manufacturing facility to facilitate phase 3 and commercial production of products such as our off-the-shelf virus-specific T cells. And another point, I would have to say that when we announced we were getting into the field of generating these COVID-2 specific T cell therapies, we in fact were approached by a number of other contract manufacturing facilities really seeking to get involved in this fight. So I think there's general buy-in of the need to move with speed and to facilitate companies that are bringing forward technology that can be effective. Just to talk separately about the virus-specific T cells themselves, as I mentioned, this is an off-the-shelf therapy generated from healthy donors who have, in fact, themselves been able to clear the virus on their own. So we're prospectively generating these virus-specific T cells in advance and then banking them for off-the-shelf use. And so with that, and with our ability to be able to accommodate multiple patients in advance, it really turns out to be a very robust way to deliver a product to patients, including at the volume currently needed to deal with this pandemic.
Thank you. And moving on to another challenge, cash flow and resources. So I wonder, George, could you talk about your role within Bio coordinating the member companies' response to the outbreak?
Sure. Look, okay. There are so many companies in the biotech industry who have either I'm sorry? Oh, okay. So many companies in the biotech industry that have some technology that would be useful for the development of therapeutics. Okay. So they have technology or therapeutics or some kind of diagnostic or approach to a vaccine, but don't have everything they need to bring that all the way to the market. And so that could be lacking in some technology. They have some promising compound, but they don't have access to the screening systems. They don't have access to a BSL-3 lab. They don't have access to, COVID-2 spike protein, whatever it is that they're lacking. And of course, these are largely in bio pre-commercial companies who have finite resources. So in many cases, they're lacking funding. And so part of Bio's mission is to facilitate the discovery and development of diagnostics and therapeutics and vaccines. And so part of that is bringing companies together so that they can have mutual discussions of what companies need and what other companies have to offer and how to match those up in appropriate ways. So, and that was initiated. There was a bio-sponsored webinar for member companies last week to try and get some of that conversation started. There's a website that's up now and is being refined that is kind of like a Angie's List for biotech companies. So you can, post what you have and post what you need and try and match that with what other companies have posted. And those discussions are just starting. But hopefully, Bio can be helpful in that regard. The other is, of course, funding and how do you get money to pay for this work. There is a lot of money that the government has allocated now that is going to various government agencies. And so the other thing Bio would like to do is facilitate the ability of Bio member companies to, first of all, know about those funds, know where they are, know how to apply for them, who to talk to, what the application process is like. And so, again, at that meeting and in follow-up, we had representatives from various government departments come speak. And that information on how to access, or at least apply for some of those funds is, again, on the Bio website, and Bio will continue to work with member companies to facilitate those interactions as well. So I think those are two of the I mean, those are the needs, both technology and capital that many companies need. So hopefully bio can't provide the capital or the technology, but can facilitate getting companies together with those who have what they need.
Thank you. And one of the other challenges, I would think, after you've developed your treatment, you've tested it, you've proved it works, what are some things you need to keep in mind as you approach approval and then rolling this out on a global basis? Maybe Z, you want to take this one?
Yeah, I mean, I think that's really a big question. I think some of the challenges, I don't.
Z, you're cutting out again.
Just want to make sure you can.
Yeah, the audio is cutting in and out.
Right, can you hear me?
I think we might have lost.
Okay, all right. So I do not know what else do I need to do.
We can hear you kind of now.
You're okay now, Xi. We can hear you now that your video's on. Okay.
Okay. Does anyone
All right, we
else
seem
want
to have
take
lost
that?
Xi. Yeah, look, your question is, what do you have to think about as you near approval? Well, drug supply is one. That has to be available, and that has to be obviously ready before you probably even begin your phase 3 trial. So that's one issue, but we've already addressed that. Look, the question of how you get these drugs available to patients around the world is a major issue. I don't think any of the companies here, certainly not Vir, and I suspect not Allogene or G, or Brie have the capability to get their drugs marketed throughout the world on their own. We'll have to have partners, and that, I guess, that could no different from other types of biotech programs. You could have one global pharma company that has commercial capabilities around the world, or we could have geographic partners in different parts of the world. And those discussions need to be underway now. So again, so you don't lose time once in the case that your drug actually works, you don't lose time getting it. So you need it.
And so, but just if I can add, I really think that the immediate issue that we need to address is the current crisis, right? So there's a need for speed at the moment to be able to bring forward these therapies as fast as possible. And I think that requires a certain amount of capital, but also a commitment to try to get therapies into patients as soon as possible. And certainly within AlloVir, we are really cognizant of the need for speed. And in fact, we're hoping to be dosing patients later this year. Again, this is in fact facilitated by the fact that we do have a platform technology that lends itself to being customized to deal with the current issue. So I think that's it. It's going to be, as George pointed out, once we get this early evidence of safety and activity, then we move forward as fast as possible to be able to roll this out on a global scale. And I think we just don't think about manufacturing to meet the now and to meet the phase 1, phase 2, phase 3 clinical trials, but thinking on a global scale. And certainly at AlloVir, that's what we're thinking on, to be able to accommodate the full extent of the patient population that could benefit from this therapy.
So I think, Z, we have you back with audio but not video. Would you like to weigh in on that question?
No, I don't have much to add. I agree. This is one of the area that we need to focus on and address this when the time has come.
Great, thank you. And so I've actually got an audience question, which ties in really well with what I had prepared, which is, how does this outbreak and your work targeting this new coronavirus affect the timeline for your other pipeline products? And specifically from the audience member, the question is, what are some problems facing biopharma companies when you're prioritizing drug products such as those needed for this pandemic response? So anyone who wants to jump in on that can.
Well, so let me comment first of all. Our company is all about the use of virus-specific T cells, and we're committed to serving an immunocompromised patient population where the need is great and where these infections are devastating. And in fact, some of our developmental programs have indeed received RMAT and PRIME designation. The magnitude of the problem and the potential benefit of the therapy is acknowledged. And so there is alignment, I think, across regulators, across the medical field and within Alavir of the need for speed here. But all that said, I mean, we acknowledge, and COVID certainly fits within the immediate need. And so, we want to advance all these therapies in parallel because there's urgent need across the board. We had to make a decision. I think we would prioritize for the short term activity in our COVID program to move as fast as possible with the immediate and urgent need that there is.
I think the impact for us is somewhat limited because our study are relatively. Sorry, George, go ahead.
No, sorry. Go ahead, G. There's a delay here, so go ahead. You talk, and then I'll come in afterwards. Go ahead.
Oh, okay. All right. Thank you. And I was really talking about the impact for us is relatively limited because our early stage and also our studies are relatively small and regional. So this gave us some ability to kind of watch the space and kind of change our strategy a little bit in terms of which country, which site we're going to go. Obviously, if you have a global study involving 30 countries, over 100 sites, I would imagine this will be a significant impact on your ability to recruit and complete those studies. So we clearly are fortunate in that case where our study is small and more regional, and we try to leverage our flexibility to move more patients, more enrollment to different countries to minimize the impact.
Yeah, I agree with that. Look, we at Vir, we have clinical programs going on for hepatitis B, for flu, for HIV vaccine, and we were starting additional clinical trials in the first half of this year. Those were the pre-COVID plans. I think most of those trials are impacted, either delayed or postponed, and that's not unique to Vir. I think that's happening across the industry. You could see announcements from BMS and Lilly and GSK, Merck, that trials are being delayed or suspended. Because it's not just a question of being able to recruit patients, which is certainly impacted, but it's an ability of the sites to maintain the rigor of the trial, to make sure you have adequate data quality so that you don't jeopardize the trial with poor data. So many clinical programs, certainly at Vir and across the industry, are just delayed. Nothing to do about that. So it's not the work on COVID that is impacting other programs. We can do all of those. It is the impact that the COVID epidemic is having on the ability of sites to recruit patients and conduct high-quality clinical trials. That varies, as G said, from site to site, region to region, and the impacts will be felt differently for different trials depending on the number of sites, where those sites are, and the timing of the epidemic. So it's a little bit up in the air right now.
Thank you. And somewhat related, I just got another audience question that talks about, as the number of treatments in development for COVID-19 increases, will there be competition to recruit patients to those clinical trials? And does the industry have a mechanism to prioritize trials or match patients to trials? I don't know if we can answer that second question, but maybe the first one definitely.
Maybe I can help on this one. Is that okay?
All right. I guess I could take this.
What about Z, then
ahead,
George?
Z.
Yes. Yeah, sorry about that. Obviously, this is something we need to watch very carefully. I think the experience that we have seen in China, and taught us something that we need to be prepared. I think for those of you who have been watching how the clinical investigation and playing out in China over the last 2 months, and literally from January 23rd to end of March, and there are more than 500 clinical investigations, and where these are all very well intended, but the net consequences is that it actually prevent some of the more promising agent to be investigated because the competition and because there's so many desperate attempt to find new agents. So for us to moving forward, we absolutely need to organize this. Luckily that we don't have to look far, and there are a lot of experiences we have learned from oncology field, as well as our approach to address Ebola diseases, where the master protocol can be designed and flexibly allowing multiple agents to be investigated at the same time in a more coordinated fashion. And it's really my hope to think about how we're going to leverage this and going forward so that we can have a centralized process, allow multiple agents to be investigated. I know that WHO has already put forward a master protocol known as solidarity trials. And I think there may be other master protocol to be developed and put forward. So clearly, this is something that we have to watch, given the enormous amount of activity in our industry. And it's not that hard to imagine that maybe 10 vaccines come up very quickly, and 10 neutralizing antibody, and 10 small molecule that will be repurposed for this. So we definitely need to work on this together.
I think she's absolutely right. And looking to oncology is actually a good analogy. There are so many drugs in development for oncology now. They're not all tested head together in the same trials, but the methodology used to test those trials has converged over the years, and the trials have a very similar design. The same will be true here. Hopefully the WHO will come out with a master protocol, and all the trials can be designed according to that protocol. I think the idea of having multiple drugs in the same trial, so you can actually compare them head to head and get direct comparative data and understand which ones offer the different kinds of benefits and risks, I think would be great. Of course, that presupposes that those drugs are all ready for clinical testing at about the same time. And I think in the real world, there are so many drugs coming forward now that they won't all be ready at the same time. And so probably the best we'll be able to do is to agree on what the appropriate protocol is, what kind of data we're going to collect. And so there'll be, with the caveat that it's always dangerous to compare data from different trials run at different times. You'd like to minimize the risks of being able to do that. So I think there is a danger of a chaotic situation, but hopefully, not only the WHO, but I think the NIH is thinking about common trials and common trial design. So hopefully there'll be some order brought into the process.
Thank you. Anne, I see you nodding. Is there anything you want to add before we move on?
No, I mean, I'm in alignment with the other two speakers. I think at the moment there's so many therapies coming online, and I do think the only other thing I would say is not all of these therapies are designed to function at the same time in the disease state, right? So, for example, we're hoping to intervene with our therapy in immunocompromised patients at particularly high risk of developing acute disease. There are preventative agents. And so I think when we start to parse these out and look at these different agents and when they're targeted for use, maybe we can start doing these head-to-head comparisons that George alludes to within the same clinical trial to look at the safety and the activity profile in as aligned a manner as possible.
Thank you. Just a reminder to the audience that you can still submit your questions using the Q&A tab to the left of your screen. I have another couple of questions I'd like to get to after I ask my next couple prepared questions. So one of them is, what is different about this current pandemic, this COVID-19 pandemic, compared to previous outbreaks that we faced? And part of that is, what might outbreak response look like in the future?
So
I think everybody's waiting here. Go ahead, G. I see your picture. Oh, go ahead, Anne.
Yeah, so I let me just first answer. You know, for me, when I look at this COVID-19, the thing that makes it really challenging to work against is the fact that it hides in plain sight for so long. So people can be infected, asymptomatic, and spreading the infection. And we've all heard the numbers and how fast the problem escalates. How is that different? So there's a speed and an infectivity component to the COVID outbreak. It looks a little bit different to others, but I mean, personally speaking, you know, I've lived a lot of my scientific career in academia, and specifically in the world of virus-specific T cells. And what I can tell you is that there's a real, in my opinion, waxing and waning in interest in looking at viruses and being poised and being ready to deal with pandemics such as we're currently trying to deal with. And because the funding environment is inconsistent, and there are times of abundance, and then there are times when it's very challenging to get funding to do new research using targeted therapies. I think what I would like to see going forward is a real commitment to be able to support the really broad infrastructure that is needed to ensure that we remain poised and ready to deal with what comes next. Because as we're seeing now, there's a need for preventative agents, there's a need for good diagnostics, rapid screening strategies, good therapies, and then that can be safely deployed on a large scale. And so it's really maintaining, I suppose, the support around keeping good scientists, good clinicians, people who are very invested in maintaining that kind of wealth of knowledge that's needed to deal with such an event as we're currently dealing with, to keep those in the virus-specific space. And so that would be something I'd like to see going forward, is a real true commitment, and not to get distracted by other diseases, but to maintain this focus on a need for readiness.
George, you want to go next?
Sure. Look, I think what distinguishes this from other pandemics is the infectivity of the virus. What Anne said, people can walk around for days asymptomatic but infectious, infecting lots of people. There are super spreaders who appear to be people who infect, tens of other people, not 2 or 3 other people. Whether they have exceptionally high titers and are asymptomatic, I don't know what distinguishes those patients. So, that results in almost 1 million patients worldwide now having been diagnosed. And I'm sure the actual number is much higher than that because the rate of testing has been so low in many countries. So, the total number of patients with SARS was 8,000. And we're well over 800,000 now for COVID-2. So way more infectious. What I hope we can learn from this is, we were thrown a warning about what coronaviruses could do with SARS. Everybody got excited about SARS for a while, and then the epidemic died down and the enthusiasm waned. We got a gentle warning with MERS. Again, there was some excitement for a while, which then waned. And now we got hit over the head with COVID-2. And so what I hope can come out of this is the realization that even after this pandemic subsides, that there will be a need and there will be other pandemics that hit us. And whether those are coronavirus or flu or flaviviruses or rhinoviruses, there are a limited number of viral families that can, let's say, that have the highest potential to lead to pandemics like this one. We should be prepared, and society should be willing to take the resources that it will take to do what we can to be prepared for the next coronavirus outbreak, for the next flu outbreak, or for Zika or dengue or whatever the next viral outbreak is. So if there's any good to come out of this, it's certainly not much good, but if there's any good, I hope it finally sinks in on people that we need to take some resources and be prepared for pandemics.
And Z, do you have anything to add?
Well, I totally agree with George and on both points. Maybe if there's anything that I would add, I think is the realization, how much we have learned about this outbreak, and the realization that now this outbreak is already connecting both the northern as well as the southern hemisphere. So it's more likely than ever that this will come back and do the next season. So it may stick around for another year or two. So we need to really absolutely focus on, doing everything we can and to be prepared for the next, outbreak, or at least the major outbreak that's going to come to us in the near future. So we really need to work on that in the short term. That is different from the SARS and MERS.
Thank you. And just before we wrap up, I have a couple last questions from the audience. It looks like they're probably both for George. One of them is, what is the website that is the Angie's List for bio?
Good question. I don't remember I don't have the URL for that website, but if you go to Bio's website, okay, you can be directed there.
Okay, great. Thank you. And the second question actually goes back to what you were talking about at the very beginning about Veer's work. It was, how does increasing antibody half-life affect long-term immunity?
Well, maybe I wasn't clear. Infecting half-life doesn't impact long-term immunity. Maybe the antibodies can provide protection for months instead of weeks, but not years, right? There is a separate FC modification that comes out of some academic work, Jeff Ravitch at Rockefeller, which indicates that at least in animals, in both tumor models and now in a flu model, that mutation can both increase near-term potency of the antibodies and also lead to the generation of long-lasting T-cell responses. That is due to an alteration in the FC region, which alters the way the antibodies bind to stimulatory FC receptors. So these are called FC gamma 2A and 3A. As well as, and binds more tightly to those, the antibody, and it eliminates binding to a inhibitory FC receptor, gamma 2B, and so it shifts the balance and causes both macrophages, monocytes to be activated to a greater extent, and leads to activation of dendritic cells, which then present the antigens to T cells. So all antibodies have the ability to do that to some extent, but of course, they're a tightly controlled process with both negative and positive controls on it. And so by stimulating the positive controls and inhibiting the negative controls, you can cause those antibodies to basically do what all antibodies do, but do it in a more potent way.
Thank you, George. So that was our final question. We did get a lot of great questions today, and we couldn't get to them all. I see a couple still in my queue, so we'll try our best to get back to everyone who submitted a question personally after this webinar. I'd like to thank everybody for attending this virtual roundtable and submitting these questions. I'd also like to thank our speakers for participating and Patheon by Thermo Fisher Scientific for sponsoring the webinar. The session has been recorded and you'll be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.